students, today we will read about the transforming principle. Okay, as you know that previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan and numerous other scientists had narrowed the search to the chromosome, clear located in the nucleus. But the question is remains same, that what is the genetic material? Clear, but till that time no one was able to give the answer about the genetic material. Okay, so we know that chromosome is located in the nucleus, but till that time the discovery was not given by any scientist related with the transforming material or genetic material. So what was the experiment given by Frederick Griffith and how it led the milestone in the discovery of DNA? Clear? That thing we will discuss now. So what is the transforming principle? In 1928, Frederick Griffith has given and has shown a series of experiments, clear, which is related with two things he had kept in his mind. First thing about one bacteria and second thing is an object, mean animal, clear. So first thing what he observed, what he has taken for his experiment is Streptococcus pneumoniae. Streptococcus pneumoniae, which this bacterium responsible for pneumonia in mice. Clear? So second thing what he had taken for his experiment, it's about the mice. First thing one bacteria, second thing is about mice. These two things were used by Frederick Griffiths. Clear? So in his experiment what he did, we will discuss it. Clear? So in his experiment a living organism had changed in physical form. How it has changed? So this thing we will discuss. Clear? So this bacteria, which bacteria? Streptococcus pneumoniae, are grown on a culture plate, and some produce smooth shiny colonies and some produce rough colonies. Clear? So you can observe in this pic also S strain, R strain. Okay, so firstly I will tell you what is the meaning of S strain and what is the meaning of R strain. So according to Griffith experiment, he used two forms of Streptococcus pneumoniae. What are these two forms? One is smooth type of Streptococcus pneumoniae and second is of rough type of Streptopneumoniae. Clear? Streptococcus pneumoniae. So what is the meaning of smooth type of this bacteria? A smooth type of bacteria means this bacteria is having the capsule present around their body. Clear? So this type of bacterium having capsule around their body, but in case of rough they are not having any kind of capsule. If capsule is not present, that's why they are known as the rough type of Streptococcus pneumoniae. Okay, now I hope that this much is clear, that two types of Streptococcus pneumoniae were used by Griffith. First is of smooth type, second is of rough type. Second thing, the smooth and rough type of Streptococcus pneumoniae are having different strain type. Strain type means they can be strained with a different type of dyes. So smooth type of Streptococcus pneumoniae can be S1 strain type, S2 strain type, S3 strain type, okay, and so on. Like rough one is, it can be R1 smooth type, R2 smooth type. Clear? Are you getting my point? Mean that he used different type of Streptococcus pneumoniae. Okay, smooth and rough. Why is it smooth? Because capsule present around in their body, that's why it's smooth. And if a capsule is not present, that's why it's known as the rough type of Streptococcus pneumoniae. Next point, we should we know that Streptococcus, if they are two types, so they are having the different characteristic. So according to Griffith, why he used that particular type of smooth and rough type of Streptococcus pneumoniae? Because a smooth type of Streptococcus pneumoniae is a virulent type. It's a virulent and pathogenic. What is the meaning of pathogenic? Means it can cause the disease. Okay, and the rough type of Streptococcus pneumoniae, rough type of is non-virulent and non-pathogenic. So one more important point we should keep in our mind, that Griffith used one more characteristic about them. He used S third type of strain. S means smooth one, okay, and R2 strain of rough Streptococcus pneumoniae. Clear? So when he performed experiment according to this method. Okay, so in first case you can see that different 4 type of pattern he used to experiment to perform his experiment. Clear? So in first condition you can observe that S strain is virulent. S means S3 strain. Clear? It's virulent and live as strain. Virulent means live as strain smooth Streptococcus pneumoniae. When he injected that material inside the mice, okay, that time mice died. It is expected, as we know that S strain or S third strain or smooth type of Streptococcus pneumoniae is virulent and pathogenic. So definitely the result is expected, the mice gets to be died. In second condition you can see R type of strain is non-pathogenic and non-virulent. So when this material is injected inside the mice, that time mice is able to survive. Clear? So mouse live, no S strain is present. Clear? In the second condition you can see no S strain is present. In first condition you can observe that S strain is present because S strain is pathogenic as well as virulent. In third condition you can say if this S strain 1 Streptococcus pneumoniae is heated. So at the heating time, you know, after heating we can observe that the bacteria gets to be killed. Clear? Bacteria can be killed by heating. So he observed that heat killed S strain bacteria when injected into the mice. That time it did not kill it. This type of strain not kill mice. That time mice survived. So mouses lives no living S strain. Why is it so? Because S strain is heated, and because of heating this bacteria gets to be killed. In fourth condition you can see that is the most important, and this is only led the base of this genetic material. Clear? You can see what he did. He did S strain. Which one S strain? Heated at S strain material. Okay, and he used second thing R2 strain bacteria. He just mixed this both the material and then injected it into the mice. In this case R is a live one but S is the heat killed one. Clear? So that time you can see on screen also, and you can observe that in this case, as it is expected that mice should be live, but due to this S strain material the mice gets to be died. Clear? So after this he concluded that R strain bacteria had somehow been transferred by the heat killed S strain bacteria. Means R is live, so R is not having any kind of content. Clear? But S is S. Which one? S? Heat killed S strain bacteria is having something which is transformed R strain also. Means S strain के पास कुछ ऐसा है जो R strain को चेंज कर दे रहा है S strain में. Clear? So R strain to synthesize a smooth polysaccharide coat and become virulent. एस स्ट्रेन ने ऐसा कुछ कर रहा है कि आर स्ट्रेन अपने बाहर एक कैप्सूल बना रहा है और आर स्ट्रेन एस स्ट्रेन में कन्वर्ट हो जा रहा है, बिकॉज यू नो दैट वन डिफरेंस इज ओनली बिटवीन आर स्ट्रेन एंड एस स्ट्रेन इज प्रेजेंस ऑफ कैप्सूल और एब्सेंस ऑफ कैप्सूल. Okay, so are you getting my point? So but this is only the base of the genetic material. Something is transferred from S to R, but that time he was not able to define it properly. Clear? So after his experiment, after 16 years of Griffith experiment, 3 scientists has given more information about it. What are the 3 scientists? Okay, so we will read about the another experiment, that is biochemical characterization of transforming principle. Clear? So Avery, MacLeod and McCarthy in 1933 to 44 they worked to determine the biochemical nature of transforming principle. Clear? So the base was given by Griffith, so उनके काम को आगे बढ़ाया. Clear? So he just did work on transforming principle in Griffith experiment. Clear? One thing that they observed that they purified biochemicals from the heat killed S cells to see which one could transform live R cells into S cell. Means they did experiment on the basis of Griffith experiment, that whatever the things, which things is presence there, which is only changing the S cells, which is only changing the R cell to S cell. Clear? So what thing is present in heat killed S cells which changing R cell to S cell? They discovered that DNA alone from S bacteria caused R bacteria to become transformed. Okay, they have only given the experiment. They remove lipids and sugars from a solution of heat killed S cells. Protein, RNA and DNA remain. Clear? So they use 3 things. Just keep this in your mind. So what they did, they just taken out the tissue from the mice. Which mice? Which died. Clear? Smooth strained heat killed bacteria and R2 type of live bacteria, when injected inside the mice, that time mice died. We know that experiment was given by Griffith. But how they continued the same experiment? Okay, they just taken out the tissue from the mice and they analyzed it. Clear? They analyzed it. What they analyze? They analyze inside the tissue live S3 type is present. So they were surprised that how S3 type is present inside the mice tissue. Clear? So something in S3, heat killed S3 bacteria, which can change R2 life into S3 type. Clear? For this this experiment, they did one more experiment related with proteinase. Clear? Protease. Protein digesting enzyme is protease. Clear? They use RNase and they use DNase. Clear? So in first experiment, what they did, they add in heat killed S cells. Okay, in heat killed S cells, they just add up protease enzyme. That time you can see that S cell is appear. Add R cell also, S cells are appear. So what it means? It means that protein digesting enzyme not responsible for transformation, because same cells are present in this case. Second case, RNAs are also not responsible for any kind of change. But here you can see that S cells are not present. If S cells are not present, it means that these S cells are digested by DNAs. Mean that this treatment proves that molecule which was responsible for result is DNA. Clear? Only DNA can cause transformation. They conclude that DNA is the hereditary material, but not all biologists were convinced at that time. Clear? Because that time it was very difficult to convince other scientists also, because scientific base and analysis was not up to the mark at that time. They were not having but a good biotech lab and all. Clear? So that time convince to other scientists, biologists, was very difficult task. Clear? So you can see here, so all at you can see here in the both the cases R cell and S cells appear. Clear? But in last case no S cell appear. It means that the DNA is responsible for the changing of R2 type, R2 life type to S3 type. Okay, so I hope that much is clear. If you have any question, you can send me your question. Thank you, children.